Hello everyone and welcome back to our video series on respiration. I've just come back from a jog. My whole body feels really tired now. I hope that you guys are also taking a break here and there and not just sitting in front of your screen. We all need a good break sometimes to help our learning. So I'm ready to carry on now. In the last video we talked about running. We talked about the changes that take place in our body when we run. And in particular about our muscle cells and whether they carry out aerobic respiration or anaerobic respiration under different circumstances. Now in this video, we're just going to end off our first set of notes by talking about how we study respiration. So this video is just going to be a very short introduction to that, but you really need to go and read your textbook as well to understand the experiments that are used to study respiration. Hopefully this video gives you the key so that you can understand the textbook a bit better. Now often in the O levels they'll give you questions where there's an experimental setup and you're meant to understand how that experimental setup studies respiration. Now something that I find very helpful is that when you approach these questions, you need to have one key idea in your mind, and that key idea is the equation for aerobic respiration. Now the equation of aerobic respiration tells you the reactants and the products of respiration. And this is very helpful, right? Because this equation tells us that if aerobic respiration is taking place, then we will detect oxygen being used up, right? If aerobic respiration is taking place, we will detect carbon dioxide being produced. If aerobic respiration is taking place, then we will detect some energy being produced, right? And some of this energy that's produced is lost as heat energy. Okay, so all of this can be used to prove that an organism is respiring aerobically, right? So if we sense oxygen being used up, carbon dioxide being produced, or heat energy being produced, it kind of indicates or suggests or supports the conclusion that an organism is respiring aerobically. So in the rest of this video, we'll see some examples of that kind of experiment, and you'll see what I mean. So let's begin with a very interesting chemical called hydrogen carbonate. Now hydrogen carbonate is interesting because it changes color as the concentration of carbon dioxide around it changes. So at the normal atmospheric concentration of carbon dioxide, hydrogen carbonate is red in color, right? Hydrogen carbonate solution, red in color. Now as the carbon dioxide concentration increases from atmospheric levels, then hydrogen carbonate indicator changes from red to yellow color. Now this is also because at high CO2 levels, the solution becomes acidic. Now on the other hand, if the concentration of carbon dioxide decreases from atmospheric levels, then hydrogen carbonate will change from red to purple color. Right? So low CO2 purple color. So this is an interesting observation and it can actually be used to study respiration. So how can hydrogen carbonate be used to study respiration? Now here's an example of one way. So here I have two test tubes, right? And both are similar. Both have hydrogen carbonate indicator at the bottom. And you'll notice what is the color of hydrogen carbonate. It's in both cases it's red in color, right? Because this is the normal atmospheric concentration of carbon dioxide. Now over here I have in one test tube an insect placed on a support. And my experiment is asking, is this insect respiring aerobically? So what do you think will happen to the color of the hydrogen carbonate indicator after some time? Now think about it and then start the video again. Alright, so hopefully you thought about it and you came to some sort of conclusion, and maybe you're thinking, yeah, okay, so the insect here will be respiring, right? It'll be respiring aerobically. So aerobic respiration. And as a result, it will be producing CO2, right? Produce CO2. And that CO2 is going to dissolve in the hydrogen carbonate indicator, and it's going to change the color of this to yellow. So that's exactly what is observed. After some time, the control hydrogen carbonate indicator remains red because there's no CO2 being produced, and in the setup with the insect, right, it respires aerobically, so you'll notice that the color of this changes to yellow after some time. Right, so this is one way to study respiration, the production of CO2. Now here's a second experiment that can be used to study respiration. Now I want you to just pause the video at this point and just try to look at this setup for a while and try to make sense of it, try to answer the questions and make sense of it for yourself before you go on. So ask yourself, what is this experiment doing? How does it work? So pause it and have a think for yourself first. Okay, so how does this experiment work? Well, the first thing is that you got to catch the idea of where the flow of air is. Okay, so there's a suction pump here sucking air this way. So that causes air to be taken in from this side. Air comes in from the atmosphere. Now the air that comes in will pass through potassium hydroxide solution, and then that air will be bubbled into the lime water, and then there'll be more movement of air this way. Right, so the germinating seeds will receive some sort of air, and the air from there will then be passed into the container here containing lime water. So again, whatever comes out here will be tested in the lime water, and then finally the suction pump works this way. So what you realize that the question here says what will happen if flask D after some time, and why? And hopefully you realize that after some time, the lime water here will turn cloudy. And why is that? That's because the germinating seeds are respiring, right, aerobically. So the germinating seeds will produce carbon dioxide, right, which will be passing through this way into flask D, and this carbon dioxide will dissolve into the lime water and turn it cloudy, right? That's simple. So what is this container here for, container B? Well, in order to understand container B, we got to look at container A. So atmospheric air is sucked into this pipe and comes into container A. Container A contains potassium hydroxide, and the important thing you need to remember about potassium hydroxide is that it absorbs carbon dioxide. So what's happening is atmospheric air is coming in, and the carbon dioxide inside it is being removed. So whatever air comes into container B does not contain carbon dioxide. So there's no carbon dioxide in the air that comes in. So the bubbling here, the lime water here should remain clear. And that is to make sure that whatever air comes into the germinating seeds does not have any carbon dioxide in the first place. So that whatever carbon dioxide is produced and detected in flask D comes from the germinating seeds alone, right? It doesn't come from outside. That means the carbon dioxide that's detected in flask D came from respiration alone and from nowhere else. Now for the other two questions, you can think about it in your own time. So I hope you get a rough idea of how this apparatus works. Now here's another experiment. Again, I would like you to pause the video at this point and just try to understand it for yourself first before carrying on. Okay, so how does this experiment work? Well, the first thing you realize is that there are thermometers. So it's something about measuring temperature. And you remember that if aerobic respiration is taking place, so go back to your equation, then you will produce energy, of which some of it is heat, right? So that means that if aerobic respiration is taking place, we should notice a rise in temperature. Now you also notice that there's two setups here. One, we have peas soaked in water. The other one, we have boiled peas. So predict, which one will be respiring? Well, your prediction should be that the boiled peas will have no respiration, right? Because they are boiled and they're probably dead and better for eating. Whereas the peas soaked in water, these ones will be respiring aerobically, so aerobic respiration. So over time, what you will notice is that there's an increase in temperature here, but not here, because respiration is taking place here. Now, the last detail that you might have noticed is this antiseptic. So what is the antiseptic for? Well, if we know antiseptic is used to kill things, right? Kill microorganisms. So why is it important in this case to kill microorganisms like bacteria and other things? And you'd be right to think that the reason for this antiseptic is to make sure that there's no other organisms which could be respiring aerobically. So imagine if we didn't kill the bacteria, right? And there was some bacteria inside here. That means they're boiled peas, but there's still some bacteria that somehow survived the boiling, right? Then those bacteria will be respiring and it would cause some change in temperature. Then our experiment wouldn't be accurate anymore, right? It wouldn't be accurate because we're measuring the respiration not of the peas, but of the microorganisms. So that's why we need this antiseptic to kill off whatever other organisms that could be producing heat energy through respiration. Okay, good job for persevering till now. Here's one last experiment to look at before we're done. So here I want you again to pause the video and to think about this experiment. Appreciate it for yourself, try to understand it before you carry on. Alright, so how does this experiment work? First, you realize that there is a animal that's placed inside this flask. Next, you realize there's something here that is soaked in potassium hydroxide solution. So immediately your bell should ring that this absorbs carbon dioxide. Okay, and finally you realize here there's a water drop up here. And the question asks you in which direction does the water drop move and why. Now, hopefully you thought a bit about it, and the answer is that the water drop will actually move in this direction, right? Toward the flask. And why is that? Now remember what's happening in respiration, right? Respiration is glucose plus oxygen gives me carbon dioxide and water. Oxygen is being used up, right? So as this grasshopper respires, the stained water drop will move to the left because the oxygen inside this flask is being used up. Right, so as the oxygen here is being used up, this stained water drop will move to the left, and we can measure the consumption of oxygen by the small animal. Now, what is the potassium hydroxide solution for then? Well, this is here because in respiration, carbon dioxide is also produced. So I want to absorb that carbon dioxide here. What happens if I don't absorb this carbon dioxide? If I don't absorb this carbon dioxide, then some carbon dioxide gas will produce, and that will cause the water drop to move in the other direction. So I don't want that to interfere with my experiment, because what I'm really interested in is measuring how much oxygen is used up. I don't want to have carbon dioxide interfere, so I absorb it into here, into potassium hydroxide, making sure that any change in this water drop position is only due to oxygen being used up. Hopefully that helps to clarify things. That's really the end of the video. I hope that it will give you some idea of how we look at these sort of experiments. I'll see you for the next set of notes in the next video.